All right. Hi, everyone. Thanks for joining us today. We are very excited to be here today with one of our customers, PrecisionProfile. They have done some amazing work around cancer research and really using data preparation in accelerating the cycle of cancer research. So we're excited to have them on this webcast with us. My name is Farnaz Erfan. I'm the head of product marketing here at PacSada. And with me today, I have Dave Parkhill. He is the co-founder and CEO of PrecisionProfile. Dave, prior to PrecisionProfile, Dave was part of Hitachi Consulting, focusing on healthcare and life sciences industries, and specifically on big data implementations and technologies. Dave has had more than 25 years of experience leading large system implementations of IBM Global Services. He was also the former VP and Chief Architect of Global Healthcare Exchange, the CIO and CTO of Virgin Mobile USA, and founder of 2 startups. Dave, glad to have you today with us.
Thank you, Farnaz.
Great. Okay, so the agenda for today is I'm going to go through a couple of slides just for those of you who are not as familiar with PacSada, but we will spend majority of the time with Dave to really go through their journey at PrecisionProfile. So a lot of you have seen this little statistic on this slide here. The point is that the majority of time of any data project is spent on data preparation. And that data project could be anything. It could be your analytics and reporting type of projects. It could be your data science, or it could simply be an application migration type of project. And the reason for it is that we all know data in its raw form is not shaped in the form that it should be. It requires to be cleaned and contextualized. And traditionally, this work has been done within the IT team. So IT has had the tools, they've had the expertise and the coding skills to be able to prepare the data and turn that over to business users. But the problem with that is there's a lot of back and forth. There's a lot of iteration that happens as part of this cycle with the data going from IT to the recipients of the data that are sitting in the line of business. And as a result, majority or a significant amount of time is spent in this kind of iteration and endless mode of figuring out how the data should be cleaned, how the data should be prepared. A lot of companies struggle with this. However, we've had quite a bit of companies that we've worked with that have really changed the dynamic of this cycle. And one of those companies is PrecisionProfile, who are, who is with us today. The other companies, we have examples of are HMS. HMS is healthcare services company. What they do is that they do sift through claim data for their customers. Their customers being providers and Medicare and Medicaid type of organizations where they're trying to figure out fraudulent claims and reverse that into new revenue for their customers. They were struggling with really bringing claim data together from different sources as claims come in different formats and form factors, and that was a cycle on average of over 300 days, and with PacSada, they have been able to shrink that cycle to 90 days. Oath is a company that some of you are perhaps familiar with them. They are a merger of Yahoo and AOL, and as part of their merger, they went through an application migration scenario, and that project was estimated to be a 22-month project. But with the use of self-service data prep, they were able to accelerate that into 3 months. And then last but not least is Standard Chartered Bank, which among many different use cases, they have reduced compliance reporting. And the majority of that was because they did the data prep for compliance reporting. They did that within the line of business. And that's really at the heart of why we are here today, at the heart of what PacSada is here to empower. We're really here to empower the business analysts, the subject matter experts, and the citizen data scientists to be able to prepare the data on their own instead of really waiting for IT to do that. And the reason for that is that, these folks are are most familiar with the context of the data. You're a subject matter expert, and therefore you shouldn't really wait for IT to prepare your data. And Dave will touch on some of these examples in his conversation, where we've been able to, or PrecisionProfile has been able to enable oncologists and researchers to prepare the data on their own. And then the other reason is that in any organization, there's just not enough resources or technical resources to go around. So the idea is to really empower the ones that are, are most familiar with the data. And with that, I'll turn it over to you, Dave, to take us through your journey with PrecisionProfile and what you guys have been doing.
Thank you, Farnaz. It's a pleasure to be here. I appreciate the opportunity to tell our story. So if you could advance the slide, let me just give a quick overview of what PrecisionProfile is all about. We are about accelerating insights for cancer treatment. And we do this for both patients and oncologists with the goal of extending cancer patients' lives. And we do this by providing self-service analytics to the researchers and physicians that are looking at genomic data. The thing to keep in mind here is that cancer is now known to be driven by genomic alterations or mutations. These alterations and mutations can come from a number of sources. They can be hereditary, they can be environmental, or they can just be random mutations. But as a result of this knowledge, the whole availability of genomic data and precision medicine is changing the conversation about cancer treatment from tissue of origin such as bladder, breast, or prostate to the underlying mutations. So if you have lung cancer, a physician may speak about what's called an EGFR mutation, or if you have bladder cancer, there may be something called an ALK1 mutation. So as a result of this, we're now able to understand what's happening at the molecular level. So if you could advance to the next slide, what we see here is that there's this data explosion occurring, and it's occurring as a legitimate big data problem. We deal with files that are terabytes in size, and it's probably the biggest big data problem out there. If you think about, say, the population of the United States and everybody getting their genome sequenced, that's going to be a lot of data. So the next slide kind of illustrates why this is happening. The cost per sequencing a whole genome has dropped faster than what one would expect if it were following Moore's law. So you can see that as of 2015, the cost of doing a whole genome sequence for a single person is about $1,000. It used to be, as you can see back in 2008, it used to be about $10 million. So it's readily available. The next slide shows a little bit about kind of the accumulative collection of genomic data. And if you look at the red line, which is the historical growth rate, by 2025, we will have a zettabyte of genomic data collectively across the industry and across the healthcare practices. So this is a massive data issue. There are no standards for genomic data, or very few actually. And then there's the problem of integration of the genomic data with EMR data. And so we use PacSada in this context to help us get a handle on the massive data that we have as a result of the inexpensive sequencing. So the next slide talks a little bit about what our product is. Won't spend too much time on this, but just to give you some context, we're a SaaS-based, cloud-based SaaS service. We incorporate AI and machine learning and provide visual analytics. Our goal for researchers is to help them do accelerated drug discovery. For oncologists, we want to enable them to get to faster treatment plans. And for patients, we want to get to a precision treatment sooner rather than later. So how do we do this? Well, the next slide shows the data that we ingest. And so you can see that we bring in a number of different types of data, the key ones being the cancer characteristics and the patient history. We also have a database of previously sequenced patients, which comes from the National Institutes of Health, the Cancer Genome Atlas. And so we ingest that data, bring it into our workbench, give the physician and the researcher the ability to visualize the data. And the goal is to get the patient the first treatment as the right treatment. So the next slide then kind of illustrates how we use PacSada. So we bring in a set of files called the variant call format files. These files are the output of the genomic sequencing process. So the high-level process is that somebody will either take a saliva sample or take a tumor sample or take a blood sample. That will be sent to a sequencing lab, and then the output of that is really a data file, this variant call format file. We also then add mutation significance, which is an algorithm calculated against each mutation to determine its importance. And then we bring in clinical information, such as patient medical records, previous treatments, whether they perhaps have had chemotherapy or whether they have perhaps had radiation therapy. We bring that all in, we ingest it, we shape it, we enrich it and classify it, and then combine other data sources in as well, clean it and publish it to a data lake. So our goal is to be able to handle pretty much any type of data that we see, regardless of what its structure is and regardless of where it comes from. The next slide illustrates a key problem that PacSada solves for us. So these VCF files for a cohort of 400 or 500 patients, that can be well over 100 gigabytes. And so these VCF file records, if you look at the top blue bar there, that's about 18 million rows for that cohort. The structure of those rows is that they're organized by gene ID and patient ID, and then have a repeating field of the variant calls. So the variant calls are in fact the actual mutations that the sequencing process has identified. What we need to do is take that repeating field or that nested field and transform it to something that we can manipulate in an SQL format. So we take the variant calls that are separated by a vertical bar and turn them into several variant records for each gene and patient combination. This would be extremely tedious any other way but PacSada. And so the PacSada split function enables us to extract these multiple entries and put them in the structure that we can use in a relational form. The next slide talks a little bit about the kinds of data that we ingest. We have cancer moonshot data, which you may be familiar with. The cancer moonshot, it was sponsored by Vice President Joe Biden, and it's now turned into the All of Us initiative, sponsored by the NIH. We bring in data from cancer clinics, EMR data, patient data. There's a new set of companies out there that are actually paying patients for their genomic records, Nebula Genomics out of Boston and Luna DNA. If you get your genome sequenced, either for cancer or for just the general genomic sequencing, such as 23andMe, Nebula's idea is that you should get paid for that data, and we can ingest that data as well. We're also working with claims data through a company called Definitive Healthcare. We also work with sharing networks. Orion and Open are organizations of clinics and academics that have agreed to share cancer patient data. And then, of course, we bring in the genomic profiles using the VCF files, and we get those from the molecular diagnostic source or pre-existing sources such as the Cancer Genome Atlas. The next slide really just sums up what we can do. Dan Theodorescu is one of our sponsors and founders. He's an oncologist and a researcher. And the goal of our platform is enabling him to be more informed as in his role as an oncologist and a researcher. The challenge here is complicated because the data is not standardized. It's in several different places. There are a number of various tools and annotations that can be applied to the data. And so we're really simplifying Dan's work for him in that we get the data in a place where he can look at it. Another key factor is that in the research community, researchers in this space can easily spend 60% of their time just wrangling data. Our goal is to get them back to the lab, not have them become part-time computer scientists, and let them focus on what they really know how to do, rather than trying to take data that's in difficult structures and various locations and merge it all together, because that really is not their key capability. PacSada gives us the ability to give them that capability and get them back to actually doing research. So with that, I will turn it back to Farnaz. Thank you for your attention and your time.
Great. Thank you so much, Dave. That was fantastic. And I like the reference of a part-time computer science, part-time healer. Thank you. That was amazing. Okay, so we're going to go to some questions. If anyone from the line has, if you guys have any questions, please go ahead and submit that in the Q&A box, and we will read those out and answer them as we go through them. All right, so one of the questions is about the data types and formats that PrecisionProfile is bringing into PacSada. Dave, you covered quite a bit of different set of data sources. Would you talk a little bit more about them in terms of what is the type of data and structure of data that you're bringing in?
Sure. The key data that we bring in is this variant call format file. And as I mentioned, this is the output of the sequencing process. So a tumor or a sample will get sequenced, and the data resulting from that identifies variations for that tumor or that sample versus what's called the human genome reference. And this human genome reference was developed as part of the big project 10, 15 years ago to get a map of the human genome. And because we have 3 billion base pairs in our DNA, that is, if you think of the double-strand helix, there are 3 billion of those things in everybody's DNA. So getting a variant call that says, you know, this person has this variation at this location, they might have hundreds of those. And the question becomes one of which ones are significant, which ones are possibly related to cancer. So sorting through all that data is a big challenge. But then we also come to the problem of most of these things are called out as a location. So the genome map is numbered essentially. But the genes also have names. And so when we bring in a VCF file, we get the number reference. We take that number reference and map it to an ensemble name so that we know which gene that references. And this helps the researchers kind of understand, okay, semantically, what gene am I looking at here, and what kind of mutation is this potentially. We also bring in data from a number of other sources. Clinivar is one of them, and there are probably at least a dozen tools that genomic researchers use, and PacSada enables us to bring in that data in a very easy and efficient manner.
Great. Thank you so much for explaining that. It's a lot of data blending and parsing. I hear from
Right.
what you're going through. It's a lot of different file formats. Fantastic. Another question just came in that's asking, have you faced any specific challenges in bringing in Unicode characters in column values, as you have different combinations of files to bring into PacSada? Unicode characters. I'm not sure if, David, you have run across that as one of your types of data sources that you bring in, and if you can talk through it. If not, we could definitely get back to this individual after the call.
You know, as far as I know, we haven't hit that, but I'm happy to ask our technical team and see if that's an issue they've run into. But as far as I know, I've not heard of that as an issue.
Okay, great. Sounds good. Wonderful. And the same person is asking that in terms of the experience in working with unstructured data in PacSada and nested structure in PacSada. I'm not sure if any of the files you bring in are more like flat files, delimited, and you certainly have covered a lot of that, Dave, from a semi-structure similar to JSON files. I can answer that question, and if you like to chime in, that would be great too. The experience in PacSada for semi-structure files when your file is nested is a tabular flat format. So regardless of your original file type, you're able to really bring that into PacSada. PacSada parses that into a table format. So it's a lot easier to explore and discover and understand anomalies as compared to coding around a nested structure type of file. Anything you'd like to add to that, Dave?
No, I would say that that's a very helpful feature for us because sometimes we do see nested structures in a flat file. We sometimes actually see structured files, and the fact that PacSada can solve that for us without us having to either write a script or do something else really makes the process go faster.
Okay, perfect. Thank you. Someone else is asking, is there an ability to ingest and translate unstructured data such as MRI, X-ray, doctor's notes, and memos into PacSada? So as far as unstructured goes, if it's PDF files or if it's images, it's not something that PacSada can read that in, but in terms of notes, emails, and things like that, we could, you could kind of go through and parse it. You could kind of recognize characters. For example, there may be a long address value or a note, and you could say, I want to search for this pattern and kind of parse that out into a different column. So you could do some of those things in PacSada to parse your data and split that into values that you are interested in. But from a X-ray or MRI files is not something that we can do within our platform today. All right, so another question just came on in terms of the deployment models that PacSada provides. So from what, Dave, you guys are an AWS customer, correct?
Yes, that's correct. We are hosted in that way.
Hosted in AWS. So PacSada covers different deployment models. We do have our own PacSada cloud that runs in AWS, but that means that from your standpoint, you don't have to really spin up a cloud or backup or recover your work. PacSada does that for you. But if you do have your own private cloud, that being AWS or Azure or anything else, you could use the virtual private cloud mode of PacSada and run that within your environment that way. Dave, do you want to share a little bit about your deployment model further?
Yeah, we've actually found it very easy to use, and the big advantage of a cloud solution is that we can turn PacSada on when we need it and turn it off when we don't. Mm-hmm. And it really doesn't take much time at all to get it up and running. It requires a little bit of some scripting to actually get everything sequenced in the right order. But it's economically, it's fantastic for us because we can control our spend and only have PacSada running when we need it.
That's a great point. Great. Sounds good. So those are all the questions we had today. Really appreciate everyone joining. I wanted to leave the contact information for Dave and myself. If you guys would like to reach out directly, you're welcome to. Dave, you've kindly provided your email address. So for the folks that are interested in more of the pharmaceutical, clinical, and specific healthcare examples, please reach out to Dave. And we want to leave you with some additional resources. We have a PrecisionProfile case study that was available as part of this webcast to you as a PDF. You could download that from the webcast interface right now. There's also a case study that was featured in Dataversity with Dave as a spokesperson. And then if you're interested in the product, we have a 14-day free trial that you can kind of try out and see if this is something that will help you in your work. And hopefully this webcast was helpful to you. I would love to, I'd like to thank everyone for joining the call today and giving us your time, and also Dave, for you to be part of this webcast with us, and really appreciate your time as well.
My pleasure. Thank you.
Absolutely. Great. And with that, we are going to sign up for today and wish everyone a really, really great day.